microphage because it is a small eater, but it is a phagocytic cell. And it is the most prevalent in the bloodstream, so it's the first one to arrive. As it comes in, it will phagocytize that bacterium or pathogen, and that thing that is engulfed inside the cell or phagocytized is called a phagosome. That phagosome then is digested through the vesicles with lysosomal enzymes or digestive enzymes in it, will actually kill that bacterium or pathogen, leaving its antigens. The neutrophil then will expel through exocytosis those antigens back into the interstitial fluid, which will then enter the lymphatic system and go to the lymph node. Okay, right now, in the same instance, we have monocytes coming into that inflamed tissue, where the monocyte will then become a macrophage. The macrophage is a large eater. It will phagocytize that bacterium, creating a phagosome. It'll lysosomal digest through the bacterium or pathogen, and then leave the antigens as well. At this point is where the story changes between these two cells. This macrophage then, up here in the nucleus, we have a process where we create, from chromosome number 6, we create proteins through from messenger RNA, right? So that's how we do that. The proteins will change in a specific gene there to change the shape of that protein just a little bit, and it's a random process that changes that. That's chromosome number 6, by the way. So that then, that mRNA makes a protein, and that protein goes into the phagosome, and that phagosome presents each one of those proteins to the antigen, okay, to try and find a random find for that particular antigen. Now we're calling the antigen here a star antigen, but remember that bacterium and pathogens are they're antigenic mosaic. It means they have a lot of different antigens on their surface, okay? But in this case, we found one that's the star antigen, and then this messenger RNA, then the protein that this guy made is actually called a molecule called MHC2, which is major histocompatibility class 2, which is your immune system. It is what identifies you as you and the process for fighting things that are not you. Now, that macrophage then will attach that MHC2 molecule to this antigen, and then it will express it on the surface of the macrophage. Now, once that expression has occurred, this macrophage leaves the tissue where it's at and enters the lymphatic system and goes to the lymph node, where in the lymph node, it will start looking for a lymphocyte to make a T helper cell. It does that by taking the MHC2 class 2 molecule and the antigen and presenting it to random lymphocytes or T cells. Okay? So it does that until it finds one that antigen will then fit into that T cell receptor. Okay, so this is a T cell receptor. Now, once that antigen fits, there's also another receptor here for the MHC2 molecule. All right, and then if this antigen fits and this MHC2 molecule fits in this receptor, then that we have just made CD4 signal or first signal. Okay, so this is the first signal. Once that occurs, CD3 is stimulated, which is a cluster designation on the inside, which stimulates the nucleus. Once this is made up here, MHC2 class 2 molecules will actually start expressing interleukin 4, which will attach to interleukin 4 receptor. Interleukin 1, which will attach to interleukin 1 receptor. That's interleukin 1 receptor. And that is, interleukin is between the white blood cell communication, right? Okay. Next, we have a second signal or co-stimulation from B7, which comes from the presenting cell, which is the macrophage, which is called a presenting cell, right? B7, which then attaches to the receptor called CD28. CD28, interleukin 1 receptor, and CD3 all must be activated before the first gene inside this T helper cell can be activated. Now, all this is done, first signal, second signal, and the interleukin-1 communication, because we need to be very certain that this is actually needs to be done, because it is a very powerful process and we can start affecting ourselves. We're actually finding kind of drugs that will affect the CD3 stimulation, MHC2 molecules, as well as we're trying to control very specific portions of this so we can be much more specific on our activation of the immune system to fight diseases, cancers, and things like that. So this is really exciting stuff when you start talking about disease fighting. Okay? All right, so we have the CD3, interleukin-1 receptor, and CD28 are all activated, which then stimulates the first gene in the nucleus, which then releases a hormone called interleukin-2, which then stimulates from the second gene the receptor that is expressed on the surface of this cell, interleukin-2 receptor. This process is called autocrine stimulation. In other words, it is stimulating itself. Interleukin-2 receptor, when activated, will then stimulate the third gene, which allows this cell to then go through mitosis. As this goes through mitosis, it creates more and more and more and more cells of this very specific T helper cell that has this very randomly generated, matured in the thymus T cell. Okay? So now, this is the actual antigen that this cell will help destroy through the antibody process. Okay, so now this is kind of the end of the T helper cell as it pertains by itself. So then we have the neutrophil that released these antigens into the tissue, the interstitial tissue that then went into the lymphatic system. The lymphatic system picked it up and took it to the lymph node. In the lymph node, we have a bunch of B cells which are matured in the bone marrow. These are all random again. Okay, so we have this antigen that is floating through this lymph node. The B cell receptors on the end there. You'll notice that has no presentation here. It just has to stimulate it. But once you stimulate this B cell with this antigen, it expresses on the surface two receptors, interleukin-4 receptor. All right, and once that is stimulated, interleukin-4 receptor, then we also have interleukin-5 receptor stimulated. Interleukin-4 is what we call from this T helper cell, which is called B cell growth factor, interleukin-4, and then from the T helper cell we also have B cell differentiation factor, interleukin-5. So B cell growth factor just interleukin-4 stimulate interleukin-4 receptor, goes through and it causes, once it stimulates, causes the mitotic gene to start. Interleukin-5 actually changes the machinery inside the B cells to turn this into a plasma cell, which is the functioning B cell. It is actually what the purpose of a B cell is to make plasma cells, which then make antibodies. Okay, now, all of these antibodies are secreted from the plasma cell, huge Golgi apparatus, all that are very specific. They have the star antigen on the short chain of the antibody, which then attach to the microbes back at the area of infection, which allows for this, which we call opsonization, when it's a flag, right? Which waves at the macrophage and says, come over here and eat this one. This one needs to be eaten. It's a bad guy. Okay, so then the macrophage or the neutrophil dendritic cells or any of the other phagocytic cells will actually come over and can then destroy this bacteria, right? Or it'll activate complement where it actually just punches in, well, that is part of complement, but it'll actually just punch a hole in the plasma membrane of that cell. Okay? That was easy.
Okay.
Right?
Sure.
Piece of cake.